Hey, everybody, and welcome. We're going to get started in about 2 minutes, so sit back, relax, and enjoy some background music while we hang out and wait for everybody to join. Right, I think it is time to get started. Welcome, everybody. Happy Wednesday afternoon, evening. For those of you that are attending live, super great to have you all here. I'm David Vaillencourt, founder and CEO of the GMP Collective, and you are at the 12th episode of When Things Go Wrong. And today we have a really great show. I'm super excited to have Dr. Nandu Sarma and Pamela Epstein on, which I will introduce briefly. Just a couple of housekeeping items. First, especially if you're new to the webinar series, bring your questions. We have a great Q&A function here, thanks to Zoom. We really encourage your participation. We're here for you. This is educational, insightful, so ask your questions and we will do our best throughout the 1 hour to answer them. For the ones that we don't get to, we will look out for a follow-up email where we will cover other unanswered questions as well as provide a recap. Let's see, what other things? Just don't forget the other 11 episodes, as well as all of them as we continue to move throughout the series year over year, are accessible for free on our YouTube channel. So you can view our YouTube channel. We'll throw that link in the chat in a moment to link and share with your colleagues. With that said, let's jump in. We have a jam-packed topic today titled "The Spec Symphony: Determining Identity, Purity, Strength, and Composition." And I am so grateful to have longtime colleagues at this point, not just in the cannabis industry and dog years, but truly I've been working with Nandu. I think we've known each other since some of the early ASTM days in 2018. And Pam, I remember you at a couple of meetings in California, sitting in the audience being like, that lady is a badass, and I'm so glad that she is a force in the industry. And here we are several years later, having gotten to work together on a couple of great work items that we'll talk about today. So with that, ladies first, Pam, can you introduce yourself to the audience here?
Sure. Hi, everybody. Thanks for joining. And me and Nandu and David, when things go wrong is the best time to assess how to do them correctly, right? We learn the best from our failures. In my everyday job, I'm the Chief Legal and Regulatory Officer for Terpene Belt. Terpene Belt is in the hemp terpene space. And so we service both the cannabis, the hemp, and the traditional markets with our product. And predating that, I worked for Eden and in their cannabis arm. And before that, God, it's been a while. I had Greenwise Consulting, where I helped operators in California and Arizona navigate the complexities of getting licensure and maintaining compliance.
Thanks so much, Pam. Yeah, you have got quite the story of adventures in the industry. Nandu, sir, so great to see you. Introduce yourself, please.
Thank you. Thank you for the invitation, David, and pleasure to join you and Pamela for this conversation. I'm Nandu Sarma. I'm Director, Dietary Supplements and Herbal Medicines at US Pharmacopeia. I am a pharmacognosist, trained on the chemistry and pharmacology of botanicals. So I've been working on botanicals all my life. I have been working on cannabis since 2016, when USP wrote a stimuli article on the topic and also organized a roundtable conversation on this. I work with the Cannabis Expert Panel at USP and several stakeholders, including other standards organizations such as the ASTM, AOAC, regulators, and others. So I want to give a shout out for the American Society of Pharmacognosy, which is meeting later this week. We have a workshop on botanical drugs that I believe will also address some of the questions related to the cannabis guidance from FDA. We have a session on cannabis in that meeting. It is right here in our hometown, Rockville, Maryland, ASP, American Society of Pharmacognosy. Thank you, David.
Yeah, thanks, Nandu. And I think that really tees up the overarching theme, as Pam was mentioning as well, with when things go wrong, opportunities to learn from our mistakes. And I think recognizing the American Society of Pharmacognosy and the workshop, the annual event that you guys are hosting later this week is another perfect example of there are so many resources out there and items that we can draw on so that we don't have to make the same mistakes twice. And so, with that, Nandu, I'm going to keep you in the hot seat here to kick us off. And actually, let me take a half a step back to just go over the topic a bit further. So, when we talk about the Spec Symphony, what do specifications really mean and do? And so, think about specifications as identity, purity, strength, and composition, which is, I believe, the US Pharmacopeia's key definition. And, that plays a vital role in ensuring consumer safety and product consistency, as well as regulatory compliance. Just as members of a symphony orchestra, work together to make a harmonious performance, these specifications contribute to the overall quality and experience of any products, especially our topic, hot button issue of cannabis topics. And so with that, I want to jump in for Nandu here, with this theme of learning from lessons past. So how would you compare the current state of the cannabis industry with its lack of standards to the situation in the pharmaceutical field before the establishment of organizations like the US Pharmacopeia, which is now over 200 years old? That was a big birthday you guys celebrated last year. And further, what can we learn from the history of pharmaceutical standards to apply these lessons to address the challenges faced by the cannabis industry today?
Yeah, thank you for that, David. Cannabis is a medicine, and there was a time before 1820 when there were no standards for the drugs in the United States at that time, meaning that, if you ask for valerian tincture in the state of Arizona versus New York, you would get different things. And even within the same state, different cities, within the same city, different places, it might mean different things to different people. So there was not a consistent quality of product which was being provided. So 11 physicians from around the country, they came together in the Library of Congress in 1820, 200 years ago, and wrote this US Pharmacopeia. So, and fast forward 200 years now, for prescription drugs, because USP is official compendium and mandatory for prescription drugs for compliance with public standards, you don't see that kind of problem. When you walk to a pharmacy and get a prescription for, let's say, amoxicillin or Vicodin or any other medication, you take what the what the pharmacist gives you, and whether you go to Arizona or New York or even Australia, you get the same quality of the medicines that conforms to the same specifications. So that is an important safeguard, and so that the patient can actually get the benefit of what the doctor intended him to take for that specific illness. So those were the driving philosophies, and as a result, there are a lot of pharmacopoeial approaches which are developed over a period of time. And several of your attendees might know that USP had a monograph for cannabis from 1850s to 1940s. I'll provide a link to a Herbal Gram article that we wrote on this topic. So unfortunately, the same apologies. Safeguard is not available for cannabis at this time. You go to different parts of the city, you get different products. So these are some of the things where you can learn from history, and you can use the standard setting approaches and the available standards to help reduce the risk to the consumers, the patients.
Well said, Nandu. I think that story cannot be repeated enough. I'm sure for many people here it's the first time, but even every time I hear you share it, Nandu, it's just so relevant and resonates. Whether you think about different strains and how we're calling them, not being consistent within state marketplaces, let alone across state borders, especially if you're a medical patient that is lucky to have reciprocity and you're traveling, because states, especially on the East Coast, are not that large, and you might have family in another state. You might go on vacation, and to get a product and have a completely different experience that night or day versus the previous week is that's not medicine. That's not acceptable, and that's what we had 200 years ago that formed the USP, that caused the USP to be formed, and we just have so much to learn. So thank you for sharing that. I'll go on, Pam. I'm curious to hear your perspectives on this, and there's a couple things that come to mind, right? One is we've had cannabis around, documented history being used for over 5,000 years. But, the wide range of products available today is significantly different than what was, say, recognized by the Chinese emperor, let alone, and it's even still different than what was in the USP's monographs and the National Formulary from the 1850s to the 1940s. And so I'm curious, what's your, in light of all this evolution, how do you see this underscoring the necessity for comprehensive standards, including these specifications that address, again, that diverse array of products in the marketplace?
It's a great question, David, and it really builds off of what Nandu was saying. It's the mission critical importance of standards and what they bring to the consumer, right? When we're looking at harm reduction, when we're trying to understand the ubiquitous nature in which e-commerce allows us to both be connected and disconnected, especially as we look at the current marketplace, which is a source-based marketplace, which I'm sure is something that is vexing for USP, and that we are treating a molecule differently. We're trying to break down botany and change it through a regulatory makeup. And that creates a lot of distortion. So as an industry, we need to be good stewards of information. And right now, there's a little bit of a disservice that happens when we claim that cannabis has been used for thousands of years, right? Well, that's vaguely and arguably true, but maybe for phytocannabinoids, and in what format flower would be really the only one, or hash or something of that nature that was really used at that time. But we don't have your parents' cannabis, we don't have your grandparents' cannabis, and we certainly don't have ancient cannabis. So to draw those parallels and say that I can borrow that safety data and bring it forward is really dangerous, because then you can have opposition or concern really punch holes through a good quality argument that this should be medicine. And so when you think about that, Think about beer. Back in Mesopotamia, it was very much closer to water, but it was fermented, it was boiled, it was safer. But we would not call the beer in ancient times the beer of today, right? It would be laughable. We wouldn't make that comparison. And yet we're making that comparison with cannabis to our own detriment. And it becomes even more problematic when you look at the different modalities of consumption. When you look at concentrates and food and things where cannabis is just finding its way into all of these new outlets, which is an amazing part of the plant and how consumers interact with it, but then further deepens the need for standardization. And this is separate and apart completely from saying that cannabis that is novel or artificially converted has any relation to the cannabis that was in ancient times or even 20 years ago, 30 years ago. So we have a duty to be forthright. We have a duty to come armed with knowledge and the awareness that we need to bring more scientific validation. We need to take that anecdotal evidence and put it into practice and put dollars and spend there, because investment dollars are anchored to scalability, which is based and predicated on harm reduction and safety, that is safety and toxicology that is validated. And that's what standards can really provide to us, is that shift in the marketplace. Use the history that we have to highlight the need for standards and validation of today.
Yeah, thanks, Pam, for that. Nandu, kind of building off of that a bit, and this is a challenging question that I know you are, it's even challenging to ask as a complex question to me. So but I'm excited to hear you kind of break it down for us. But when we look at, everything from the chemistry of the plant, right? And we've got a diverse array of cannabinoids that are found naturally in the plant. Some are intoxicating, like are commonly known, delta 9 THC. Some are not. There's this symphony that they work, perhaps that they work together with the ensemble effect. But like, how do we integrate the chemistry and the biology of this product in our interaction in the body to contribute to the assessment of intoxicating cannabinoids and the establishment of standards? Like the quantification of cannabinoids using chemical systems, how does that enhance our understanding of their biological effects to ensure, what we really need to see in the marketplace, right, is this accurate, predictable, and reproducible assessments that can guide consumer safety when, eventually translates to dosing. So that's kind of a big question if you want to take a stab at breaking that down for us and sharing your perspectives.
Sure. Glad to do that. Yes. So I'll take a step back to one of the old USP monograph to put a context, and then go to the current standards which are published by USP. So if you look at the USP monograph, USP 910 in around 1940s, you will see that it was a brand new introduction of a biological assay for cannabis extract. So wherein you give certain amount of extract and look for in coordination in dogs. So it's kind of, a biological assay, functional assay. So it was a state of the art at the time. And it has its direct correlation. Like if you look at some of these, there are two of these botanical drugs approved by FDA, green tea extract, and the other one is the Croton leschleri extract, the exudate. And so there is a biological assay for that in terms of, enzyme inhibition assay. That's functional property. And that is because it is really complex in case of these Croton exudate. It's a complex one. You don't really know what is really acting there, and you use a functional assay. And compare that with cannabis. What the cannabis expert panel did at USP very early on was to kind of distill the information on what is known in terms of the biological activity, what components are known to be active, and the data is out there. A lot of data is out there, and so you have the delta 9 THC is a very dominant one. So all these different what are called strains, they are varieties, botanical varieties of the cannabis plant. So they are developed or bred in such a way that they are dominant for either the delta 9 THC, or whether they are dominant for the CBD, or maybe intermediate varieties of these. So this was the work that was done by the expert panel very early on to define what is responsible for the biological activity. They identified some of these dominant cannabinoids and the minor cannabinoids. And so right now, what we have is a very clear chromatographic pattern of the different cannabinoids in an extract. So you have an extract, you can easily run in a chromatogram. It is no longer a challenging part. It's a very standard processes for any of these established labs these days. And so you can identify which components it is. And so compared to the earlier times when it was a biological assay in dogs, you have a much more refined assay in which you know which components are there. You know that delta 9 THC is the prominent one, which has these intoxicating effect. Of course, the definition of that itself is still being debated, but you have that you can quantify that accurately. And you can similarly quantify the delta 8 THC, you can quantify the CBN and others very accurately, and you can qualify these different extracts based on these profiles very accurately. So that is the current state of understanding to which we can apply the technology and enhance our understanding. So what the panel did in the recent times was to write a journal chapter 1568 on how to appropriately conduct a clinical study using these different chromatographic approaches and other approaches. I'll share the link after this presentation, and that's a resource for the community. And it really defines what chemicals they are. So it connects the biology and the biological activity with the chemistry. So that's the beauty of the current state of knowledge.
And I think that's a really important state to connect the dots on, in terms of being able to look at a complex botanical plant and use like fingerprinting and I don't know that biomarkers is the right word at this in here, but like, yeah, with these fingerprints and the dominant ratios of known cannabinoids to categorize, right, and classify as chemotypes and chemobars, right, where we have the 3 different types, and we can use that as an approach to say, here's what, if you're using type 1, type 2, or type 3, those are going to have elicit different effects, and we have to figure out a way to break it down. And that's exactly what you guys have done for us, based on pioneering a review of a lot of literature and data points from the marketplace. Yeah, thanks, Nandu. I'll add, and then we've got a question that came in that I'll throw in there in a moment. Thank you. Keep throwing them in there, folks, if you have questions. I want to jump into purity, though. I think this lends us into a talk of purity, and there's a difference in purity between a botanical substance and an API, a single molecule, which we're well familiar with on the pharmaceutical world. But we've got these products out there, delta 8 products or delta 9 hemp derived products. Like, what's the purity of them, and why does that matter? Specifically, these are not found in a lot of these, like delta 8, are not found in nature, so they're not phytocannabinoids. And they may be synthesized in a lab. So how do the impurities affect and impact the quality and safety and consistency of these products, and what measures can be implemented to ensure effective impurity control in the industry? Nandu, I'll cue you up for that one.
Sure. So specifically talking about the delta 8 THC, of course, if we take a step back, these cannabinoids can come from the plant, so they are inherently complex and inherently variable depending on several factors, and also depending on whether what solvent they are extracted with, how they are refined, whether they are decarboxylated or not, whether they are winterized or not, whether they are distillate, and so on. So there are a lot of processes to which they may be subjected to. Depending on that, the composition and the nature of that material would vary. And what comes along with it, depending on, again, the reaction conditions from the plant. So we typically call them as related compounds. So that can also vary. For example, USP recently proposed a monograph for the CBD as an active pharmaceutical ingredient, and the CBD coming from natural sources. So the related components would be different than if we were to develop a monograph for CBD as from the synthetic origin. The precursors are different, the reaction conditions are different, the catalysts are different, and so on. So that would be a different one. So these things have to be controlled appropriately. And also, when we generically talk about CBD and delta 8 THC, what conjures to the mind is that, okay, this is highly pure substance that we are talking about, like salt, that comes to the mind, like pure crystalline thing. And typically when we define a CBD article, it is not less than 98.0% purity and not more than 2.0%. All the related compounds, water, residue on ignition, and everything combined is not more than 2%. So you have a highly pure expectation. So you see the name CBD is connected with the quality attributes in that monograph. So if we talk about the delta 8 THC, what normally comes to the mind, a consumer might think he is getting a delta 8 THC, but unfortunately, all the delta 8 THC in the market has to come from synthetic sources because it is inherently at a negligible levels, at less than 0.1% levels in the plant material. And so obviously the multiple milligram levels and hundreds of milligrams that people are the companies are marketing have to come from the synthetic sources. So this depending on the synthetic reaction conditions, depending on what is the origin for this material, depending on the catalyst used, temperature used, it can contain not only the degradants, but also reaction intermediates and so on. So the need for controlling the impurities, that's a question, is really serious. And to just put perspectives to that, we worked closely with Dr. El Soli's labs in Mississippi, and we communicated a paper for publication. We identified 14 impurities and developed analytical methods for resolving these impurities and quantified these impurities in 21 vape products. And what we found was that all of them exceed more than 0.3% of the delta 9 THC, so they are no longer hemp, meeting the definition. And the content might vary depending on the product. It may be 50% of what is claimed on the label, or maybe 300% of what is claimed on the label. And the impurity profile varies from 7% to 43% of the impurities. So you have a lot of, and the impurities are also not consistent across these different products. So there are concerns of these impurities, and that needs to be controlled.
So I wrote down 4 things that came out of that, and that, I mean, God, yeah, I have so many thoughts that I would love. We could muse about this specific topic for probably hours on end, because it's just that vast and complex. You covered things such as thinking about the source material, how it's processed, what catalysts, what grade of catalysts are being used, and these are all considerations that get way into the technical details into a batch record in a in a mature marketplace is where they should be reflected. That defines and helps a chemist understand what compounds, what impurities may be there, so that we can then use that to say we should look for those. And we don't have that in the regulated marketplace, even, let alone the unregulated marketplace. And so the paper that you discussed, which I remember Pam and I were actually together at in Oxford at an amazing annual conference that, thanks to you, Nandu, with the FDA and natural botanical marketplaces, the International Conference on the Science of Botanicals, where a member from the El Soli lab had a poster presentation on that paper right before it came out, and we were blown away with some of the points you just highlighted there. First, these commercial products that are claiming to be Delta 8, which if you extract that to what the common consumer recognizes that to mean, it means, oh, this should be a pure Delta 8 product like any other pure drug. It's not. It not only violates the Farm Bill's requirements of less than 0.3% Delta 9 THC, but it has over a dozen impurities that are found. They're not consistent, and the impurities range from did I get that right? 7 to 43%. Mm-hmm. That's significant. That's far and above the 2% number. I could go on, but I think Pam, this tees you up to I don't even know if this is a question, but where do we even begin with the gaps in the marketplace regarding purity from your perspective?
Well, I think you got to step back, right? First, we have to unify this plant again, because the body has no idea if you are ingesting a cannabinoid from hemp, from marijuana, or from yeast or orange peels, right? Your body's going to react to that cannabinoid the same way. So from that premise, when we look at our current regulatory schematic, in the regulated space, we at least have good quality control, quality assurance testing. Now, that testing tests for microbials and heavy metals and a lot of these other kind of impurities that Nandu was talking about, but it still really focuses on potency as its driver. So you have to, as Nandu said, know what you're going to look for in order to assess those potencies. So within the regulated space, we're better. We've got a baseline. We do still have more work to do, especially when we start to want to regulate around novel cannabinoids or chemically converted cannabinoids. I think most people don't necessarily know that CBN, typically that you are consuming commercialized, has been converted oftentimes from CBD. They want to speed up that natural degradation process of that phytocannabinoid in order to commercialize it. So we should be paying attention if there's Delta 8 in the regulated marketplace around testing specifically for impurities on that side of the line. When you look at hemp, it's really different, and it is problematic because we test for potency, right? All of the regulations around hemp are around one molecule, Delta 9 THC, and whether or not it's 0.3% on a dry weight function. Now that happens in the field. We test only hemp federally when it is in the field pre-harvest. There is no additional requirement unless a state has specifically instituted that requirement. And there's no track and trace. So there's no linkage between a harvest batch, what Nandu was talking about with the impurities that potentially may be in the field that are drawn down into these products. It is, a really risky endeavor there for consumers. So we really want to impart this idea that I like to think of as 2 P's and an E. You have to understand the potency and the purity in order to do good enforcement, right? You have to be testing, because without the requirement to test, there are good hemp operators out there. I do not want to deny them that. There are some operators who are doing the exact thing that they need to do, but your system is only as strong as the weakest link. And when we don't have standards as a base around purity, you see exactly what was coming out of the paper that Nandu was talking about at A. Soli's lab. It is that when you just pull these things at random off of the shelf or on e-commerce, you are finding that they have both impurities and excessive amounts of Delta 9 or other intoxicants that are getting to consumers, and they don't know that that's what they're consuming. And that's problematic, because if somebody has a reaction, we don't even know how to treat it because we're not even sure what they've taken, because it's not necessarily the cannabis or the cannabinoid that's harming them. It's the process. So we really need to understand and appreciate the certainty that can come from standardization in process, and what catalysts you can use for this conversion and what catalyst you're going to test for at the end to make sure that that catalyst has been neutralized throughout that process or removed, as well as the outputting cannabinoid, what you put in, and the process testing for purity and then potency, so that you can get both of sides of that coin, which is necessary for the regulations to protect consumers out in the marketplace. So there's really a big void in dealing with purity in a holistic respect. So as we like strive, and everybody will hear me say this, as we strive towards unification of the plant, then we can really grapple with how do we best understand these cannabinoids in their raw phytocannabinoid stage, and as they become cannabis concentrates, and how those can become final form products, because there should be testing at each of those pivotal points. More importantly, even than what is in the field, is what that concentration is for purity and what that outputting final form product that goes to market is.
So I want to add, and then we're going to go to what I would really like as a question that came in. First, I'm going to share another link. So I put 2 links in the chat. The first one that I put in was the paper that Nandu was referencing that was published in April. It is behind a paywall, but I definitely encourage you to read it, and I will give you a sneak preview to know that, God, I'll add, there's so many links. We'll put it in the recap email, but I will be releasing with a group a podcast in early August where we're actually discussing this paper. So we'll be diving into some of the highlights and kind of breaking it down, because it is a very technical paper. That said, the second link is a useful one that that was brought up in the Q&A that I have not read in a few years. It is a great paper, Cannabis Ontologies, that was published by a gentleman by the name of Kenzie Ribelay. Oh my God, I get his last name wrong every time, but he's out in the EU, and put together concepts for how we think about it. And it is a very, again, another technical paper that dives into the different sources of abstention and the different outputs, right, of product types. And it really speaks to what Pam and Nandu have been talking about, of the source material varies if your inputs vary, and then your process and the the processes and the solvents and other catalysts that are used during the work and process, that has an effect. And then what comes out at the end is much different if it's an edible, if it's a inhalable product, if it's a topical product. We have lots of considerations that you really need to dive into, and that's no different than any other supply chain, whether it's paper, there's newspaper, there's this paper, right? There's paper that's 100 weight, 120 weight, comes from trees. Does it matter what tree it comes from? Those are the things that are processed in the supply chain that have been described in great detail with standards in order to have a functional marketplace. And that's no different than here in the cannabis industry. So with that, I want to go to this question that was brought up.
David, could I just clarify one
please?
Quick
Yes.
note? The General Chapter 1568 is behind a registration wall. So if someone is looking for that, and if it says registration is needed, it's a free registration. And we invite public comments on that until the end of this month. And even after that, we welcome that, but we will compile the comments and then consult our panel, our expert panel. So it's free, but behind a registration wall.
Awesome. Yeah, thank you for that, Nandu. So yeah, it just, as Nandu said, to reiterate the link there in the webinar chat, when it pops up to a registration, it seems like a paywall. It's not, just register, and that allows you, if I'm correct, Nandu, to now be active so that future chapters that are published, whether they're cannabis or otherwise, to provide public comment.
Yeah, it's a single sign-on, yes. And we will soon publish the Herbal Medicines Compendium monograph proposal, the CBD proposal. So all these things would be using the same sign-on.
Awesome. Yeah, so many resources, right, that we've barely even touched upon yet, but we will get to in a few moments. So, kind of with that, right, the question that came up was, looking at leadership in the industry and, being largely uneducated on, important word here, risks, right? the risks related to the inadequate purity standards for product and industry raw material suppliers. So the question is, how do you both, I'll put you both on the hot seat, whoever wants to go first, is the best way, being the best way to push forward with applying risk mitigation at this level of industry maturity. So again, the industry where we lack standards, it's all over the place. State marketplaces are different. We've got big operators, small operators, people that are largely trying to do the right thing, but there's a major education gap around the risks, these unknown unknowns. How do we address this, recognizing the level of industry maturity is not quite there? Whoever wants to take it first.
I can take it from a legal
Go for it, Pam.
Right. As a lawyer, I look at this both as an opportunity for improvement, right? There's always a need for education, but a lot of this can come through maturity of supply chain agreements, right? Whether or not you have regulations telling you to do something, you can self-impose better standards, or you can put them into your supply chain agreements. Just for example, Terpene Belt Farms, we sell terpenes. Right? They are non-cannabinoid, non-detectable cannabinoids in our product, and we sell them into regulated cannabis markets from California. We pick the California quality assurance testing metrics because they tend to be the most stringent, but then we're also tracking what other states are doing so that we are compiling the most robust way to demonstrate to our customer who is going to incorporate it into their end use product that our raw ingredient input is safe, right? That we're testing it for these things. And that can build into better education, even for your customers to be looking at scaling up their own testing metrics, right? What can diversify you? Because when you're talking about a nascent industry that is always looking for ways to grapple market share, to bring consumers, convert them back into the regulated supply chain, because we have problems with pricing, with additional, taxation models. You want to talk to people about ROI and how this is going to increase their bottom line to invest in this type of testing. It is because you can say that to a consumer. It's a differentiator right now. What we're talking about is making it a standard, but before it's a standard, it's something that you can differentiate your process and your product by, if you're B2B, to your other B2B operators, that I am providing you something that is even more of a validated thing to put into your final form product. And if you're B2C, it gives you a way to transact with a consumer in a sea of choice right now, why your product is elevated. And I think that's one way that we can really start to infuse the benefit of bringing these things to the forefront, because I can understand, I represent, operators in California as the president of the California Cannabis Industry Association. I know that it is incredibly difficult, that the industry is suffering death by 1,000 cuts, from everywhere. You have to anchor it, I think, this discussion specifically to how it's a good ROI, how it's a good investment in your company. And then that permeates out into saying, now we need standardization. There's this momentum and movement, and it makes it an easier conversation for regulators, but never miss that opportunity for education, for starting the dialogue, because the best way to get to yes is to understand every single no that you're going to get along the way. It's exactly what David's talking about. When things go wrong, right? You're going to have this conversation multiple times over, find different ways to make it impactful, to make it beneficial. And so that would be sort of my take from an investment legal standpoint. I'm sure Nandu is going to have a nice compendium to that from a science standpoint.
Yeah, it's exactly extending what you said, Pamela. It's like, doing the right thing. Doing the right thing is easy said, but I think, the freedom comes with the responsibility. Like, when we are going on the road, we can't simply say that, I didn't know. You have the responsibility to drive safely because other people are involved. Likewise, I see that the industry has that onus on themselves, whether either through compliance with very well-defined state programs, or If the state program is not very well defined too, it means they are actually transferring that burden and responsibility to the industry, saying that you got to do the right thing. You can't simply say that, you didn't know. You have the responsibility to provide safe products. The products that you say claim on the label, it has to be accurate to that. You have to use scientifically valid method. You cannot simply say that, according to my test, this is 90%. And, if you use a very simple colorimetry method, like, people used for some of these extracts, and, blueberry, they used, dye and said that, yeah, this is, blueberry extract. And, you need to use the right test to test those materials quantitatively, and limits for the contaminants. So these are expectations. You either say, you're not aware of it, you're still on the hook for those things. And when the regulations define it very clearly, I think that reduces that kind of, the burden to reinvent it. When they are not very well defined, I think the burden is probably more on the manufacturers. And what I would like to really connect is that there are a lot of resources available out there from ASTM, AOAC, USP. There are so many resources available. As standard setting organization, we know very well that, developing a good chromatographic method for nice run of, the resolution of these different cannabinoids, developing the method, validating the method with respect to all these, quality attributes, accuracy, specificity, sensitivity, range, robustness, and so on, and then verifying for the particular matrix. It's a very expensive, elaborate process. You have the validated methods available out there. You have guidance available out there. So there is so many resources available. I believe that those things can help reduce the burden on the manufacturers. And I think fora such as this, the conversation, and the other avenues when people come together, the educational events, there are several avenues for getting educated.
Yep. I love it. And so I want to kind of wrap up what I've heard and add, a couple things that came to mind as I was listening to both of you. First, education. I mean, kudos to everybody who attends our webinars. I trust that you find value and appreciate it, and continue to spread that out. Learn about things that you don't know about, and open your mind and establish that culture of continuous improvement. A culture of quality. Establish those in your institutions, whether you're a regulator that has authority and has the option to say, hey, go to this conference and learn, or go, conduct research, or take continuing education credits. Make sure that education and professional development is a key part of your organization. And that's one key way to do this. The other part that comes in is, the regulations, remember, that represents an absolute minimum standard, right? Regardless of what business you're in or vertical you're in, that is the bare minimum. And so step one, be compliant with the bare minimum. Then use the standards that exist. As Nandu, you mentioned, the beauty of standards, and not all standards are created equal, but as you mentioned, ASTM, AOAC, USP are the three primary examples in the United States actively engaged in cannabis standards development. Those are recognized. These are institutions that are a century to 2 centuries old, that are used and trusted by government, used in court and law, are used by manufacturers and supply chain agreements every day, because a bunch of nerds, as I like to say, get together, really smart people, and bring their diverse talents and lenses to the table, and they hash this out. And that represents so much intellectual capital, that when you say, I'm referring to this standard, when somebody asks, how are you going above and beyond compliance and having safe products, say, I'm referring to this standard, which reflects all that intellectual capital that gives you something to stand on above and beyond, and so much other research that you could never do yourself. Which gets into the concept of risk assessments, right? I mean, just doing a risk assessment, and that is a really challenging concept. There is so much training and education around it. But if you break it down into brainstorming, hey, Pam and Nandu, let's sit and think about what could go wrong. Let's brainstorm the many ways one thing could go wrong when I'm building my deck this weekend. Okay, let's think through that and come up with some things and come up with plans on how to mitigate that. That's risk assessments at its foundation. So doing that in your organization is also key. And then, asking those questions. And I had one other thing, but I don't want to add too many more cans of worms. So I think that's hopefully enough to help wrap this all together with questions. And it's really about that continuous improvement, right? Incremental wins are key. You don't have to bite off the whole apple, but if you take the USP's monograph and say, I'm going to add something new this month, and then next quarter I'll add a new analyte that maybe is above and beyond what my state requires, that's continuous improvement. And that is really valuable to show. With that, we've got 10 minutes left, so I'm going to get into our wrap up and recap. And so I've got a final question for Nandu and then for Pam, if that works for everybody. So Nandu, there are significant gaps in the industry when it comes to standards, but as we've been saying, existing resources. Just last month alone, you and I crossed paths 3 times, I realized as I was preparing for this webinar, twice in person at the beginning with ASTM's Committee Week, followed up by a global webinar with key stakeholders. We had Australia, Thailand, South Africa, and representation from the World Health Organization there, talking about medical cannabis. And then we both attended and spoke at the Cannabis Regulators Association stakeholder event on the value and the resources of public standards. So, recognizing that, many state agencies especially are under-resourced and their ability to protect consumers and patients. What resources and initiatives currently exist? Like, expand a bit on the general chapter. I know there was a paper in 2020 that turned into a herbal medicines monograph. Yeah, what does the USP have for resources to folks? And I'll share some links while you do that.
Thank you, David. And I really acknowledge all your contributions and support for many things that we did together. And that could not have been done by anyone alone. So I really appreciate that. I also wanted to give a shout out for the work that was done in December. That was a global workshop on the cannabis quality, engaging the regulators, the standards groups, including USP, ASTM, the Dutch Office of Medical Cannabis, and the European Pharmacopeia group, the EDQM. We had the industry panel and a testing panel, a wonderful discussion over 2 days, and identified the challenges with respect to the lack of harmonization, the different requirements, and the need for harmonization. So it's going to be a continued conversation on the topic. We recently had this webinar involving Africa and Asia, and that brought together and reinforced some of these challenges. And so that will be a continued conversation. Specifically about the resources, if you look at, although there are differences between the different state requirements, you would certainly find several commonalities. For example, testing for Delta 9 THC and CBD, CBDA, THC, THC, these are common elements. Testing for some of these pesticide residues is common elements, heavy metals, common elements. You have variations within, granted, but there are several common elements. And for these common themes, after a lot of data analysis, USP published the first publication in the Journal of Natural Products. I provided a link to that publication in the chat. And so that provided the scientifically valid methods, data-based acceptance criteria, and fit-for-purpose reference standards to support the establishment of the specifications for identity, content of the cannabinoids, content of the terpenes, and limits on the contaminants. So it's a very comprehensive publication reflecting the pharmacopoeial perspectives of controlling the quality, not just one parameter, but the quality as a whole. And besides that, USP has recently published several proposals, and these are at advanced stages. So the most recent one is the Journal Chapter 1568. We invite public comments on that. That chapter is in alignment with the FDA final guidance on the topic. And the FDA final guidance reflects upon several of the USP journal chapters, but our chapter takes it a little further with the specific analytical methods, acceptance criteria, and reference standard to support those parameters. And we also published a proposed monograph for the CBD as an API. We published a HMC monographs. These are the two others which will come back for the public comments. Again, the HMC monograph is expected to appear very, very soon. The CBD monograph will take a little bit of time. Our food ingredients team published work on the hemp seed oil, hemp seed protein. We worked on the delta-8 THC, as I said. So there are a lot of resources available out there. So I encourage people to consider using the ones which are available. It is so enormous to redevelop these resources. So that would be my suggestion.
Thank you so much, Nandu. And as if that wasn't enough resources and information, Pam, you kind of teed us up well, Nandu, with the work at ASTM going on in conjunction and collaboration, and the link on the Part 1 workshop. I just want to give a double plug to that because all the PDFs are there from, Health Canada, CANRA, myself and yourself, Nandu, ASTM and USB. There's so much information there. It can be almost dizzying, but take the time. Again, education, it's there for you. Pam, of the, I think, 70 standards that are in development at ASTM, in addition to the 50 already produced, you're working on one or two hot button ones. So can you describe a bit about the work you're doing and why it matters?
First, I have to say thank you to David, because really, my first attendance at ASTM, I was galvanized to action. I would tell everybody it is single-handedly the best conference for engaged, nuanced dialogue. If you really want to get into the weeds of weed, that's the conference to go to. And when I was there, I had such insightful conversations that over time, it really started to become clear that one of the biggest things we're missing is a standard on what is an intoxicating cannabinoid, right? Because I think the question on everybody's mind is, what is non-intoxicating? Where can I put this benchmark? And the only way that you're ever going to be able to recognize what is non-intoxicating is by knowing what is intoxicating. And so we're embarking on this standard that will be sort of the first standard of many in getting us to understand and how we can communicate that to the consumer. What is an intoxicating cannabinoid? So with that, I would encourage everybody to get involved in the standard, but more importantly, what is this standard and what is not in this standard? I have learned from the ASTM and from all of these other standarding bodies that it is the scope and the scope exclusions that are your most important thing to look at. So when we're talking about this intoxicating standard, what is excluded from there is impairment. We are decoupling impairment from now and just looking at where is the potential of a cannabinoid to be intoxicating. We're not looking at synergistic effects. So we're not looking at could the entourage effect, could putting THC plus delta-8 equal a higher intoxication? That's not on the table currently. We're not looking at the method of consumption. And so those are really important scope exclusions. Now, that's not to say that once we have this standard, we are not going to have additional work items that look at defining broad spectrum and full spectrum, whether or not that can be applied from a product to a manufactured product. We're going to look at impairment. We're going to look at synergistic effects and modalities of consumption. But as my mother likes to say, how to eat an elephant, one bite at a time. This is the first bite of that apple. And it's really important because we cannot let perfect be the enemy of good. And what we need right now is not great, not perfect, but good, a good, solid standard to work from. And so that's really what we're doing at ASTM, plus this amazing classification compendium that I'm the most excited about, because it, unlike the standard where we're going to look at an assay, we're going to try to figure out how that standard should look. So as it is released, ASTM, to its voting members, we're going to be excited to get back all the negatives because that's the only way to get to yes. But this classification chart is going to function in that A until a cannabinoid can't come out of the playpen until it proves its worth, because it's going to be presumed to be intoxicating until we can validate that it's not. But we have a good amount of evidence. We have a good amount of data sets that are out there, including from USP, where we can say certain cannabinoids with certainty in the human body are known to be intoxicating. Delta 8, Delta 9, we know because federal law, federal regulation from USDA tells us that total THC, THCA, if it is intended to be consumed in a fashion where it is heated in flower, that needs to be accounted for akin to Delta 9. There are certain things like that we know. So if we can put that chart together, the hope is that both consumers and regulators and operators would know we have enough data set to clearly say these things are intoxicating, these things are not. And therefore, while we're building the standard, we have something to go to that synthesizes the available data around us with the presumption that if it doesn't show up on this chart, it's going to need to go through the standard and it should be presumed to be intoxicating. And that's how you can regulate. So I would love it to bring everybody in. I'm a convert. I went to a meeting. I never looked back. And now I look to David tirelessly for guidance on how to manage the ASTM process, but he is nothing if not the best steward of that. So I would hope that everybody checks out all of this information. There's amazing connectivity here, and the brilliance of both of these gentlemen here will get you far in understanding how to continuously improve your products so that you're putting the safest and highest quality into the marketplace.
Oh, thank you, Pam. I think, as Nandu said, and I know it's not unique to Nandu, but it's so salient with our world, it takes a village. And this is a perfect example of the ecosystem. We all bring our strengths together. And as you mentioned, Pam, ASTM is the place where anybody can show up and become a member and contribute and navigate the process to really drive these incredibly powerful outcomes. And if you're, while you wait excitedly for Louisville, Kentucky, where we'll be next in person in January, there's a virtual symposia. I just put the link in there. Be free. It's a 3-day event. I believe the CDC is speaking. There's USDA. There's multiple academic researchers, all to talk about contaminants in cannabis across all spectrums of contaminants. That is in October. So come wet your palate on the type of conversations that we're having, and then be prepared for January to get yourselves to Louisville, Kentucky, to roll your sleeves up, whether it's with the intoxicating cannabinoid standard or one of the other 71s. If there's a problem in the industry that you know of, there is a solution that can be a standard. And so come to the table, join us, and contribute. And of course, use the resources, vote, contribute through the feedback process at USP. And I would finally be remiss if I did not acknowledge there's so many people to acknowledge across the 700 members. I know APA, I think APA's on the table or showed up today to our event. The American Herbal Products Association is another great resource. The Herbs of Commerce 3rd edition was just released. That is actually recognized in Congress. They've done a lot of great work to help steer the industry, both working on the USP's expert panel and at ASTM. So there's just an amazing network of resources, and I encourage you all to not be strangers, reach out, ask questions, follow up, and share this webinar with your audience and give us feedback. So if that's not enough to do, call it a day. Go enjoy the rest of your Wednesday. Thank you, everybody, for attending, and we will see you soon. Thank you so much, Nandu and Pam. This is awesome.
Thank you all. Bye-bye.
Cheers.